for COVID-19. Dr. Louis Falo, Professor and Chair of Dermatology at the University of Pittsburgh School of Medicine and UPMC, will first describe the novel way this vaccine is being delivered.
Okay. Give me a countdown.
Dr. Andrea Gambotto, Associate Professor of Surgery at the University of Pittsburgh School of Medicine, will explain how the vaccine works.
Thank you all for joining us today to hear
finally,
how
Dr.
the University
Don
of
Yealy,
Pittsburgh
our
and
Chair of
UPMC
Emergency
are
Medicine,
working
will give a
toward
brief
vaccine
update
for
on the COVID-19 testing
Dr.
and
Louis
patient care
Falo,
at
Professor and Chair of
UPMC
Dermatology at the University
First,
of Pittsburgh
we're
School
going to
of
play a
Medicine
short
and
video
UPMC
so you can see
will
what
first
makes this
describe
new
the novel way
vaccine
this
so
vaccine
remarkable.
is being delivered.
Members of the
Dr.
media,
Andrea
there's a link to a
Gambotto,
high-quality
Associate
version
Professor
of
of
this video
Surgery
in your
at the
press
University of
release
Pittsburgh
that
School
we
of Medicine
distributed
will
earlier,
explain how
and
the
everyone
vaccine
can find it on
works.
our website
And
at
finally, Dr. Don Yealy, our Chair of Emergency
upmc.com/media.
Medicine, will give a brief update on the COVID-19 testing.
So
and
for this
patient care
vaccine,
at
we're going to
UPMC hospitals.
deliver the antigen
First,
with
we're going to play
a
a
novel
short video
technology
so
that's
you can see what
referred to
makes
as a
this vaccine
dissolvable
so
microneedle
remarkable.
array. What the
Members of the
microneedle
media,
array is
there's
able
a
to
link
do
to a
is to
high-quality
deliver
version
the
of this
vaccine
video in your
antigen
press
directly
release
into
that we
those areas of
distributed
the
earlier,
skin
and
that are
everyone can
made to
find it
make
on
an
our
immune
website
response.
at
And so it results in a very potent
upmc.com/media.
vaccine. When you're thinking about how these microneedle arrays are actually used in practice, think about them as almost like a band. Novel technology. And so the microneedle array is simply applied to the skin topically. What the microneedle placed very shortly and then taken off and thrown away. And then the antigen is already delivered.
The biggest challenge is then scale up
So it results
the
in a very
production
potent
to the number that
When you're
this
thinking about
vaccine is
how
going to
these
be eventually
microneedle
needed.
arrays are actually
We have a
used in
technology
practice,
that
think
is
about them as
pretty
almost
common
like a band.
technology. It's not
And so
rocket
the
science.
microneedle array
It's a
is simply
sub-unit
applied to
vaccine,
the skin topically,
which is
pressed into place very
relatively
shortly,
easy
and
to
then
manufacture
taken off and thrown away.
is
And then the
scalable.
antigen is
We
already
think that
delivered.
if immunogenic with the
challenge
combination of the delivery system can make possible
production
candidate
to the
for
number
a pandemic
that
vaccine.
is going to be
So I think it's important to remember that this vaccine and other vaccines that are going to be emerging are really the result of a lot of work that's been done by scientists in very diverse areas, and we need to bring all those scientists together from different areas working together to be successful in this effort, and that's what's happening
can
here.
make possible candidate for a pandemic vaccine.
So I think it's important to remember that this vaccine
After our
and
experts
other
give a few
vaccines
brief
that are going
remarks,
to be
we will take
emerging
your questions.
are really the
Dr.
result
Falo.
of a lot of work
Thank you.
that's been done by scientists in
As
very
mentioned in
diverse
the
areas,
video,
and
this
we need to
vaccine
bring
is the
all those
result of a
scientists
collaboration
together from
involving
different areas
scientists
working
from
together
very
to
diverse
be
fields.
successful in this effort,
Vaccinologists,
and that's what's happening
skin
here.
biologists, bioengineers, and virologists, all coming together to work towards a common goal. That's the type of collaborative
After our
environment we
experts
have
give
here
a few
at
brief remarks, we will
UPMC.
take your questions.
This is
Dr.
also
Falo.
the same place where Dr. Jonas Salk invented the polio
As mentioned in
vaccine
the video,
nearly
this
65 years
vaccine
ago,
is the result
dropping the
of
number of
collaboration
disabling or
involving
deadly
scientists
cases from more than
from very diverse
55,000 per year to essentially none today. For vaccine delivery, our lab developed this microneedle array approach. It is a Band-Aid-like patch that has hundreds of microscopic needles made up of a sugar-like substance that rapidly dissolves in the body. Nearly 65 years ago, dropping the number of disabling vaccine is
cases
integrated
from more than
into the needles. When pressed
55,000
onto
per
the
year to
skin,
essentially
the
none
needles
today.
penetrate the outer skin layer
For vaccine
and then
delivery,
dissolve and
our
release
lab
the vaccine.
developed this microneedle array approach. It is
The
a
skin is our
Band-Aid-like
first line of
patch
defense
that has
against
hundreds of
viruses,
microscopic
bacteria,
needles,
and other harmful
made up
invaders.
of a sugar-like
Because of that,
substance that
it has
rapidly
evolved to be
dissolves
very
in the body.
efficient at mounting immune responses, which means less vaccine is needed
The vaccine is
compared to a
integrated
traditional
into
shot.
the needles.
Our
When
process
pressed
for
onto the
making this
skin,
vaccine
the
does not
needles
require
penetrate
any
the outer
overly
skin layer
complex or
and then
expensive
dissolve
equipment,
and
so
release
it is
the
very
vaccine.
scalable. At the present time in our lab,
The skin is our
one
first line
person
of
with the
defense
settlemolds
against
and
viruses,
centrifuge can
bacteria,
make
and
hundreds
other
of
harmful
microneedle
invaders.
arrays in a
Because
single
of
day.
that, it has evolved to be very efficient at mounting immune responses, which means less vaccine is needed compared to a traditional shot. Our process for making this vaccine does not require any overly complex or expensive equipment, so it is very scalable. At the present time in our lab, one person with a set of molds and a centrifuge can make hundreds of microneedle arrays in a single day. Another important feature is that unlike most current vaccines, this vaccine doesn't need to be kept frozen or refrigerated. It is shelf-stable at room temperature, like Band-Aid. That reduces shipping costs and supports vaccine distribution, particularly to less developed countries. Our next steps include beginning a clinical trial. We are currently in contact with the FDA This process normally takes months to years, but these pandemic conditions are not normal. The NIH, the FDA, and other regulatory agencies have made terrific progress in accelerating this process. Once we get approval from the FDA, we will be ready to start human safety trials. We are speaking with the FDA now and hope to move this process along as quickly as possible. Thank you. Andrea Gamboto will now tell us a little bit more about how this vaccine was developed and how it works.
Thank you. So my lab worked on vaccine for SARS coronavirus 1 during the 2003 epidemic, and then MERS coronavirus a decade later. So when the genetic sequence for SARS coronavirus 2 was published late in January this year, we were able to plug into our existing tool and rapidly produce a vaccine. So we use a genetically engineered cell to make a piece of the spike protein on the outside of SARS coronavirus 2. This is the part of the virus that the immune system sees, and when that happens, it triggers antibody production, which should protect against viral infection and COVID-19 disease. This is how annual flu shot work. The spike protein pieces are dissolved in a sugar solution and then spun into microneedle that Dr. Fellow just described. We test our microneedle vaccine in mice and found that after 2 weeks they develop antibody to the SARS coronavirus 2. Our vaccine is based on the directly delivery of simple protein antigen, which is like many of the successful vaccines that are now in use. It does not rely on the body to make the protein like some of the more experimental vaccines under development. And because we are not using the whole virus, our approach is also relatively safe. Now, Dr. Ely will give an update on COVID-19 in our region and our hospital. Dr. Ely.
Thank you very much, Dr. Fellow and Dr. Gamboto. Thank you for sharing this groundbreaking achievement. People expect excellence in care and excellence in new care capability from a world-class academic medical center, and that's what we are delivering. As a physician and as an investigator, I appreciate the rigor and quickness that got your vaccine discovery through a peer review process, and that process allows for the best insights to become true. This is the first coronavirus vaccine candidate in this pandemic to achieve this important milestone. Many of our local and regional media outlets are interested in an update in our current case counts and the UPMC experience in both testing and in care, and I'll report on those. Across the UPMC system, we've performed over 4,100 tests, and 386 have been positive. That's a positivity rate of about 9%. Over the past 10 days, in addition to previous sample collection sites and testing facilities that existed, UPMC has set up specimen collection centers in Harrisburg, in Williamsport, Erie, Altoona, Somerset, and in Jamestown, New York. In Harrisburg, we've collected 228 samples from 2 different sites. In Williamsport, we've collected 171 samples. In Erie, 243 samples. In Altoona, 147. And in Somerset, which opened just 2 days ago, we've collected 25 samples. In Jamestown, where UPMC Chautauqua is located, we opened yesterday and we collected 20 samples on the first day. Many specimens for people who were mildly symptomatic and were being sent home to self-quarantine with those symptoms are sent on to commercial labs, while the remaining samples are tested at UPMC's central laboratory. We do not have complete results back on all the tests that have been sent off, but we have many back. Right now we're seeing positivity rates that range as low as 3% in an area, but the consistent rate is between 8 and 12%. We'll know more about the positivity rate in each individual region as we gain more and more experience over the coming days and weeks. We currently have 67 inpatients that are positive for COVID-19 across the entire UPMC system. Most do not need intensive care or a ventilator. Our community surveillance testing is still not showing widespread outbreak. The research trial that you've heard about before that supports this effort originally planned to assess findings during the common flu season, and that flu season is winding down. We are using our clinical supplies for those who need care now. The data continue to reassure us that COVID-19 is not as intensely active or widespread in the communities we serve as it is in other parts of the country. The regional efforts at social distancing appear to be paying off, and we appreciate what everyone is doing to help flatten the curve. That flattening is an effort that allows us to optimize care, to learn more about the virus, and to bring us closer to therapeutics and vaccines, just like what my partners shared with you.
Thank you, doctors. And now we'll take questions from the media, all of whom are joining us remotely online from around the world. To ask a question, you'll have to go to the participants icon at the bottom of the page and press the Raise Your Hand button. That will put you in the queue. Again, go to the bottom of the page, press on the Participants button, and then press the Raise Your Hand button. If your question has been asked and you want to get out of the queue, you can press the Lower Your Hand button. I will read your name, and your line will be unmuted for you to ask your question. We'll try to call on as many different media outlets as we can in the time that we have allotted. First question comes from Paul Goff, Pittsburgh Business Times. Paul, go ahead.
Thank you, Paul, and thank you, doctors, for taking the time. Can you talk more about how your previous SARS work, which helped in the vaccine development of this particular vaccine, and how that was so critical, why that was so critical to kind of have that, maybe could I consider it a head start, as it were?
Yes, that's a good question. So the 2003 SARS-1 vaccine, the critical aspect of that study was that helped us to identify what is the target for a coronavirus vaccine. And at that time, we determined that the spike protein that now everybody uses as a target for this coronavirus is the best target for a coronavirus vaccination. The second study on the MERS vaccine, the other coronavirus that emerged a few years ago, we test different platform, and in 2014 we determined that the subunit protein vaccine was as good as other genetic systems. So those two vaccines teach us how to translate all this finding in the new SARS coronavirus 2 vaccine.
Okay, thank you. Next question comes from Mariella Busalati from Business Insider Italy. Mariella, go ahead.
Hi, thank you. Thank you for being here. I have 3 questions. The first is your spike protein working as the spike protein of the virus? I mean, replacing it? And then the second is, when do you think you will be able to test on human beings? How much time will we need? And the third one is, why are you not using mask and you are narrower than 1 meter? Sorry.
Dr. Falo.
So in our vaccine, the spike protein is actually the target for the antibodies that we hope to elicit. So the spike protein is very important for the virus to enter human cells. And our goal is to use a piece of that virus so that we're able to, a piece of that spike protein so that we are able to induce antibody responses to block that interaction so that the virus can't enter the cell. In terms of timing, we are working now with all of the regulatory agencies that are involved in approving a vaccine. They have all been tremendously helpful, and we're very confident that as soon as we gain approval, we'll be able to start the trial.
I think the other question was, why aren't we wearing masks right now? We're trying to follow best practices, and one of which is to have the interviews done at a distance, as we're doing right now. We've kept our distance. It would be difficult to answer these questions with those masks on and for you to have clarity. So what I can promise you is when we're done here now, we'll be back in our masks.
Thank you. Next question, Tom Averill from the Philadelphia Inquirer. Tom?
Hi, can you hear me all right?
We can hear you. Go ahead, Tom.
Oh, great, thank you. I seem to be represented by a little white cartoon figure, so I apologize on the visual part. But you mentioned how you are focusing on the spike protein in your vaccine. Are some vaccine approaches targeting multiple proteins so that the virus can't mutate away?
Some of the vaccine option, when you use genetic approach, not protein approach, can target multiple antigen to induce different parts of the immune system, like the T-cell part of the immune system. Our vaccine is more focused on generating antibody.
Thank you. Next question comes from David Kaplan, WTAE. David.
Hi, thank you so much for taking these questions. I have two questions, largely. Number one, can you talk about false negatives? We're hearing a little bit more about that. Dr. Levine talked about that on a state level. Is there any concern just about the test results coming back and whether or not they could be false negatives, and just talking about your concern surrounding that? And I also wanted to ask about, we're hearing about a program for the medical professionals at UPMC St. Margaret to help keep them in a hotel and kind of offer them some things to make sure that they are well taken care of. Is that happening or planning to happen at other hospitals treating patients as well?
Thanks very much for the question. So about clinical testing, false negative results are always a possibility in virtually any diagnostic test. And false negative means the test says you don't have a condition, but in fact you do have the condition or you develop the condition. We don't know the exact false negativity rate with the tests being used now. The tests that are done at UPMC are exactly the same as what's been done by both the Commonwealth and the CDC. And we believe the false negativity rate to be low. Nonetheless, most clinicians who are skilled know that if the clinical picture seems like it's a COVID-19 infection and the first test is negative, to continue the precautions and the care and watch the pattern of the disease and consider retesting. And I don't think our experience is any different. We're looking at different testing capabilities that would help augment that, to help improve upon that particular concern. Our experience isn't any different than anyone else's across the country. Secondly, we have begun opportunities for our healthcare workers to have restful places, places away from home that are nearby the care facilities that they're at. You mentioned St. Margaret's. There's one here in Oakland, too. We will continue to develop those programs. We want our workers to be safe, well-rested, and serving the patients well.
Thank you, Dr. Yuli. Next question comes from Jamie Martinez from the Tribune Review. Jamie, go ahead, please.
Hi, thanks for taking these questions. Will this vaccine be safe for all age groups and people with conditions, some of the health conditions that maybe make them more susceptible to more severe symptoms of COVID-19? And then related to that, can you explain, I think some of our readers might be concerned that when they get the flu vaccine, they feel a little sick after. Will you have symptoms of COVID-19 after getting this type of vaccine? How might that be different or maybe dispel some of that?
Thank you. That's a great question. So this vaccine is designed to be safe, in part because it delivers very low doses of antigen, so much lower doses than the traditional flu vaccine. Secondly, it delivers that antigen to a very confined space within the skin, so that it's very unlikely that we would see any type of systemic responses that have been seen with some patients in the flu vaccine. I believe that this vaccine should be effective across all age groups. As we learned with flu, it may require slightly higher doses for more elderly patients, but that has yet to be tested with this particular vaccine.
Thank you. Again, to ask a question, go to the participants icon at the bottom of the page and press the raise your hand button. That will put you in the queue. Next question comes from Eric Tishy. He emailed to us from the Post-Journal in Jamestown, New York. Typical trials for new drugs and vaccines can take up to a year or longer, though the FDA is reportedly allowing some of these to be fast-tracked. If that is the case, what is UPMC's best-case scenario in terms of time to get this test to trial?
Thank you. So that's a good question. We are not trying to be evasive in terms of giving you a specific timeline. The regulatory process is iterative, and so how long the regulatory process takes depends on the feedback that we get during the process. What I can tell you is once we have been given approval, we will be ready to go with the vaccine.
Thank you. Next question comes from Eric Poole of the Sharon Herald. Thank you. Eric?
Previous, yes, hello. I had a question about the previous preparation that enabled you to develop this vaccine. About how the work has helped you get this developed so quickly.
So the previous preparation, which was the MERS vaccine, was similar to this preparation. So we had multiple preparations that were tested in MERS, and we down-select two of them, which then we choose as a template for this vaccine. That was the down-selection that we did from the previous coronavirus to this coronavirus.
Thank you, Dr. Gamboto. Next question is from Sean Hamill of the Pittsburgh Post-Gazette. Sean, go ahead, please.
Can you hear me?
We can. Go ahead, Sean.
Okay. Dr. Gamboto, can you tell us why your research on SARS and MERS did not go beyond the animal testing stage? And also, what about your research with SARS and MERS, plus the recent findings on the COVID-19 vaccine, lead you to believe that this vaccine would be effective in humans?
Yes, that's a good question. In 2003, when SARS emerged, we were in a front line position to really translate our vaccine into clinical testing, and we were ready back then in 2003, and we proposed what happened in 2003, that SARS suddenly disappeared at the beginning of 2014. So all the funding that were allocated for translate our initial and other group initial finding on the potential vaccine candidate just disappeared. So we didn't have funding back in 2003. There was no interest. 2014. Pretty much the same story. We developed a vaccine, and actually last year we did propose to run a clinical trial. We have a proposal that was considered for funding, but at the end was not funded. So we didn't have the resource to move this vaccine beyond the preclinical experimentation. But with the MERS, we determined that the immunity that we were getting from our vaccine correlate with what one would expect to be a protective response against SARS coronavirus 2. So we believe we have selected this vaccine format because we believe that this is one of the best and easy to translate candidate vaccine.
Thank you. Next question comes from Jessica Haberly from WPXI-TV in Steubenville, Ohio. Jessica, please go ahead. Did we lose her? All right. Okay, let's go to the next one. David Winter from Penn Live. David, go ahead.
I ask this from the point of view that I think many Americans who have been paying attention to the regular news conferences with Dr. Fauci, they've been, regarding vaccine, they've been heard a lot about how important it is, but they've also been told that it's probably 12 to 18 months away. I would just like to hear what you think about what the possible timeline is in terms of how early your vaccine could get to people, and also, I mean, I'm hearing that the vaccine's going to need to get to upwards of 45 million Americans to have the real benefit. And if you could, talk about how that could play out, or how you could scale to that level if you think you can.
Yeah, thank you. That's a good question. So Dr. Fauci is exactly right. That is the time course for vaccine development. Much of that time course, and the reason there is such a gap in how short versus how long it could be, depends on how the vaccine develops through the clinical trial. So it depends on the level of responses that are seen as the trials are being done, the safety data that comes in during the course of the trials. And so for that reason, it's impossible to give a very precise timeline on when a vaccine would be ready. We are able to start the trial as soon as we have regulatory approval. In terms of scale, our vaccine platform is very conducive to scaling. And so we will be able to ramp up production using microneedle arrays very quickly because of the straightforward technology that's involved and because of the requirements for much lower doses of antigen that need to be produced for each vaccine.
Thank you. Next question, Bill O'Toole from Pittsburgh Quarterly. Bill, go ahead.
Am I coming through? Hello?
Hello?
Am I coming through?
Yes, you are.
Yeah.
Okay. Yeah, a related question. How do you see manufacturing working practically if your trials do indeed succeed? Would it just be individual labs all over the country making them? One big center? What would it look like?
Yeah, I suspect, first let me say that we haven't gotten there yet, that all of our focus right now has been getting this vaccine into clinical trials to evaluate safety as soon as we can. But the vision would be a centralized facility capable of making millions of these vaccines at a rapid rate.
Thank you. Again, to ask a question, go to the participants icon at the bottom of the page and press the raise your hand button. That will put you in the queue. I will then read your name and your line will be unmuted for you to ask your question. So just a reminder of how to get into the queue to ask questions. The next question comes from Danielle Fitzgerald of the Beaver County Times. Danielle.
Hi, everyone. Thank you so much. Yeah, I have a couple questions. I guess this might be more for Dr. Ely. But my question was, why are the number of positive tests so low? You mentioned somewhere around 10% of tests are positive. It feels kind of low for a highly contagious virus. Do you think we're testing the wrong people or using the wrong symptoms as a benchmark? And related to that as well, I heard that losing a sense of smell and taste or perhaps even are really more important identifying symptom. I don't know if that's true or not. I wanted to get clarity on that. Should we be testing folks more with those sorts of symptoms?
Those are great questions, and thanks for asking them. I don't know if 9 or 10% is low or high. We're still learning a lot about how the virus enters a community, and it doesn't always enter every community in the same fashion. You know that when you look both globally and even across the Commonwealth, that the amount of positive cases that are either symptomatic or admitted to the hospital varies dramatically across the region. What we know is we're seeing this essentially 9 or 10% positivity rate, and why that is, I couldn't speculate. What I can tell you is that our background surveillance suggests that it's not dramatically undercounting what's available out in the community, and you've heard about that research before. Your other question had to deal with, remind me now, the second question. Oh, smell and taste. Right. It is one of the unusual things about COVID-19 infection is that early on it does seem to disproportionately, for this particular infection, change people's sense of smell or taste. And taste is often related to smell, so sometimes it's hard to separate them out. And we've sent out reminders to our clinicians to look for that finding. It's not specific, but it certainly is something that should help raise your awareness about this potential. And we've seen people not only have those symptoms, but after recovery report improvement. So I do think it is part of the clinical picture, just like the respiratory symptoms, just like fever, that talented physicians, talented nurses, talented practitioners should be aware of and ask about.
Thank you. Next question comes from Fox 43 in Harrisburg. I believe it's Matt Maisel. Matt, go ahead.
Gentlemen, thanks for taking these questions. Two questions real quick. First, how long have you been working on this particular vaccine? When did you start with the testing? And also, people just in general naturally are scared during this entire process. What do you tell them who might be watching this right now or might be watching later and are skeptical when they hear something that it may still be another year plus for something to be finalized?
So yeah, we start to work on this vaccine exactly January 21st. January 21st, the sequence of SARS coronavirus 2 became available. And with that sequence information, we were able to generate quickly. It took us 7 days to generate the actual vaccine. And then short after, we immunized the animals. So it's about 8 weeks that we are into this studies, and January, the end of January.
Thank you. We'll take another question from Central PA. Carrie Pauling from northcentralpa.com. Carrie, go ahead.
Hi, thank you. I'm wondering whether or not there's a concern if there is an accelerated trial that developers could miss dangerous side effects or other negative reactions that people might have.
Thank you. That's a very good point. What you're putting out is that there's a reason that these trials take the length of time that they do. We are taking every precaution, and you'll see in all of the trials that develop over the next months that safety is our top concern. And that's why these trials are done so carefully and can take so much time.
Thank you. Again, we have time for just a few more questions, so go to your participants icon at the bottom of your page and press the raise your hand button. That will put you in the queue. Next question comes from Ted Lutz at the Kane Republican. Ted, please go ahead.
Yes, sir. My question has to do with the testing. Who should go for the test? And we live 90 miles from Erie, and that's where one of our participants did go, and they tested positive. But I see you up in Jamestown, New York, which is a lot closer. Should just anybody go to test, or what's your policy for accepting candidates for testing?
Dr. Ely.
We prioritize the order in which people get a specimen collected and sent off. And at the top of that list are people who have symptoms, and particularly people who have symptoms and require hospitalization. That's where we can be not only most effective for their care, but make sure that people around them are the most safe. One group of people that we simply don't have the capacity today to test are people who have absolutely no symptoms, but maybe have a concern about having acquired the infection. It's not that that's unimportant, and at some point in time we'll want to know what does the population that doesn't have any symptoms, how often do they have the infection? But right now that's not the highest priority. So my advice would be seek testing if you have the symptoms. Cough, fever, difficulty breathing, the smell and taste things we just talked about. And while you're doing that, make sure you remain isolated so that if you do have the infection, you haven't spread it to others.
Thank you. Next question is Oliver Morrison from Public Source. Oliver, please go ahead.
Thank you. I'm curious if the information that you're sharing in this publication is going to be available to scientists other places so that they can be testing it at the same time. And if so, is there going to be any advantage to sharing, given the public health issues, to sharing this information that might otherwise be a source of profit freely to other countries and other places so that it would be available as quickly as possible to the people who need it?
Thank you. That's an excellent point. With this current pandemic, we are all on board with everyone collaborating to attack the virus. We are all working together and sharing data very openly. The publication that's specific to our vaccine is right now available to all of the public. It's posted on the journal's website, and we are collaborating with anyone that we can help in terms of data sharing and sharing our experiences.
Thank you. Next question, Alex Wiedhoff from the Cranberry and Butler Eagle. Alex, please go ahead.
I have two questions. The first one is, do we know what level of widespread immunity we will need to have by time the vaccine will be developed? And if so, do we know how that would be able to be deployed by vaccine, or if some people will naturally have it because they will have gotten the coronavirus already? And my second question is in terms of background monitoring. Do we have recent information, data on how many people we have tested showing up? I believe this was people showing up to the ER with flu-like symptoms, just being tested for that with not necessarily identifying data associated with that.
Well, I can take the second question. The surveillance data that you're talking about that's been presented at previous press conferences is coming near its close. There have been over 3,500 patients in the western Pennsylvania area, both adults and children that have been tested. And again, it shows that beginning through the end of January, the middle of January through now, there was not an early presence, at least not widespread, that existed of the virus. And the pattern of the virus presence right now looks very much like the clinical picture that we're seeing. In other words, there's not a disconnect. And that's in a large but not exhaustive sample.
As far as the first question, so it is our expectation that as patients recover from the infection, they will have some level of immunity. Tests are being developed to specifically address that, including tests that are being developed here at UPMC. So we would expect some amount of what is described as herd immunity. The vaccine would be a supplement to that herd immunity.
Thank you, doctors. Next question, Todd Bartley from WEJS in Williamsport. Todd, go ahead.
Thank you. You had mentioned the potential of moving to a human trial. First of all, what's the timeline on moving to the human trial? How far away are we? And then how would you end up selecting the candidates to participate in the human trials?
Yes, so the timeline will be established by the regulatory process. From a clinical perspective, we're ready to go with and initiate a clinical trial as soon as we're able to meet the necessary requirements. The second part of the question was, sorry.
How are people going to be selected?
Oh. So we will be starting with healthy volunteers in the Pittsburgh area. Those will be the first cohorts of patients that enter the trial, and we will be basically looking at multiple doses during the course of the trial to find the most effective dose.
David Kaplan from WTAE has a follow-up question. David, go ahead.
Just talk a little bit about, we heard at the federal level just talking about the next 2 weeks. So big picture, are you all preparing for and or expecting some surge? And then my second follow-up question, can you talk about a little bit about the normal timing for peer review, the normal amount of time it would take to get the federal approval for a vaccine like this, and kind of, what's the normal timeline and how condensed do you all really hope that you can make this timeline to get things going in terms of clinical trials?
So the first question is, what do we think is going to happen, and when do we think the surge will become very high, and when will it hit its peak? I have no reason not to agree with some estimates that say the surge may be at its peak within 2 to 3 weeks. The truth is we don't know. There are other estimates that would suggest that it could last even longer than that. Our approach, and you've heard this before, is that we prepare for the worst, we hope for the best, but we let the experience on the ground guide what we're doing right now and how we prepare and deploy day in and day out.
So safety is the prime concern in most of these clinical trials, and that's the reason that the typical review process can take months. We're hoping with the expedited review and all the efforts that the regulatory agencies have made that we can reduce that regulatory process from months to weeks.
Okay. Thank you. Our final question comes from Jamie Martinez of the Tribune Review. Jamie, go ahead.
Hi. To just jump off of a question that was asked earlier related to testing, how would this vaccine then be deployed to the public? Would it go to hardest hit areas first? Would it make sense for it to go elsewhere first? Which areas would probably see it, be among the first to be receiving the vaccine?
So I think that when the vaccine is actually ready for widespread deployment, it would most likely be ready broadly so that we wouldn't have to ration the vaccine. I would expect that the manufacturing and scale-up would occur during the testing process so that plenty of vaccine would be available if it proved to be effective.
Okay, thank you. And that concludes the time we've allotted. Thank you all for attending. We plan to continue holding these regular updates for the media as long as there is a need. In the meantime, reporters with questions are encouraged to contact our UPMC Media Relations Department at upmc.com/media. And please follow @UPMC News on Twitter for regular updates. Thank you very much.